In this video, I'll be reviewing the slides from the Histology Atlas that are associated with the human digestive, respiratory, and lymphatic systems. So to begin with, we take a look at a series of salivary glands. There are three salivary gland tissue types that you will be examining in lab, and it's not necessary to distinguish between the three, the parotid, the submandibular, and the sublingual. Your responsibility will be to just identify that they are salivary glands, and of course to identify different structures within each gland. So as you look at the salivary glands, they look an awful lot like the pancreas that we've looked at before. Here at 40x, you can see the parotid salivary gland. And as we begin to zoom in closer, you see a series of different types of cells, as well as ducts that are there in the system. Now keep in mind that salivary glands are exocrine glands, which is why you're going to see ducts like this. Endocrine function, like what you would have seen in parts of the pancreas, did not have ducts associated with it, but the exocrine function will. Here with the salivary gland itself, you're going to see a combination of two different cell types within each of these types of salivary glands. The first is what you can see right here pretty clearly, are what are referred to as serous cells. So let's zoom in one more time to look at these. The serous cells themselves are going to have pretty rounded, dark staining nuclei. And the cytoplasm itself is going to look relatively granular in nature. So you see large pockets here of these serous cells. They do tend to pick up sometimes a purple or a pink coloring. These serous cells are what are going to be responsible for secreting more of a watery mixture of saliva. And of course down here you can compare this to the duct. This is going to have simple cuboidal epithelium, and any duct is essentially a tube with a hollow lumen inside. Keep in mind that you should not confuse these with blood vessels, because the blood vessels that we tend to look at will either be multi-layered, right? Or if you're looking at a tiny venular capillary, it'll just be a very thin layer of simple squamous epithelial cells forming the endothelium. So this is what your parotid salivary gland would look like. If you look at a submandibular salivary gland, you can see a lot of the same things. Again, here at the 40x magnification, you can see ducts and you can see pockets of cells. As we zoom in, you can see that there's a difference here in the cell types that you would see. Now, submandibular salivary glands have a relatively even mixture of the serous cells that we've already identified, right? Plus what are referred to as mucus cells. And this is what's going to produce your thicker secretions for saliva. So zooming in right here, again, you can see this comparison. While those serous cells tend to pick up that darker stain and have the nice rounded nuclei, here the mucus cells themselves tend to look a little bit lighter. I think that they look like marshmallows or clouds. And so when you compare those to serous cells, they're very easy to distinguish. The sublingual salivary glands are made predominantly of these mucus cells. So as you can see here, just looking at the slide from 40x, it already looks quite a bit lighter because of the presence of all of these mucus cells. And so you see those nice white cloud-like cells with a few of your serous cells in between. Right, as you can see right through here. So the serous cells with the darker nuclei, darker staining cytoplasm, but a mass here of mostly mucus cells. As we move on then, we begin to look at a series of hollow organs that are associated with the digestive tract. Of course, any of these that are hollow are referred to as organs of the alimentary canal. There are accessory organs of the digestive tract that we will also examine. But as you look at the alimentary canals, the first thing that you should be able to do is identify a series of 4 tunics, or layers as we refer to them as. The innermost layer lining the lumen, as you can see here, the hollow space inside the organ, is called the mucosa. This is where you find your epithelial tissue. The next layer is the submucosa. This is made up primarily of connective tissue, and it frequently has a series of glands within it. The muscularis is what's going to be made of smooth muscle. Most of your muscularis tunics in the digestive tract will have 2 different layers of muscle, a circular and a longitudinal. The stomach would have 3. And then, of course, an outer layer of either serosa or adventitia, depending on the placement of the organ. And this is going to be a connective tissue epithelial combination here. So as we look at the esophagus, we begin to see this breakdown of the tunics themselves. If we look here at the very bottom of the screen, we can see the lumen. And now the esophagus gets compressed easily, so here the lumen doesn't look super round. But you can identify right there that lining the lumen itself, you're going to see the mucosa. So really nice coloring of the mucosa right through here. The submucosa running up with connective tissue in this direction. And then the muscularis layer itself. You can see the serosa out here on the very outside. This is going to be bordered by fat, which gets included with serosa pretty frequently. One of the things that you'll see right here is those two differing layers of muscle. So of course the longitudinal layer of muscle tends to have a pretty boxed appearance, and that's because those fascicle of smooth muscle fibers are actually running toward you and away from you, so running up and down the length of the organ itself. Whereas the circular layer of muscle, you can see these nice parallel patterns that run side by side. What you're looking at in that situation is fibers that are actually going around the organ and its periphery. So with the esophagus itself, when you take a look at the mucosa that's right there, you actually see a series of 3 different layers. So of course this image is flipped upside down in reverse to what we just saw. Here you can see the muscularis, and here you can see the submucosa. The idea right here is that lining the lumen is going to be the epithelial layer. Now in the esophagus, because there's quite a bit of friction that the food is going to create along the lining here, this is stratified squamous epithelium. More layers of epithelial cells means more protection. Just deep to that, or superficial, I guess we could say, is going to be the lamina propria, so loose connective tissue running right through there. And then outside of that is going to be a tiny layer of smooth muscle called the muscularis mucosa. Now this is different from the tunic of the muscularis. The muscle there is primarily just to change the shape here of the lumen, as well as the mucosal border in itself, but it's not really propelling food in the same way the muscularis would be doing. It is important as you look at each one of these tunics to know, or each one of these organs, to know what kind of epithelium is found in the mucosa of each organ. So we start here with stratified squamous epithelium. Right, again, you can see that here zoomed into 400x, the nice layers there making it stratified, right? And squamous indicating that those cells themselves tend to flatten out. So, of course, the esophagus is going to connect to the stomach at the cardioesophageal sphincter, also called the cardiac sphincter. And at this point in time, you actually see a change in tissue type. One of the slides that you'll look at is called the esophagus-stomach junction. And in this slide, you do need to be able to identify one of 2 things. So first off, you need to be able to recognize that this space right here is the lumen of the organ, and that would make this part of the mucosa facing the lumen itself. Now, of course, the esophagus, as we saw, was going to have stratified squamous epithelium forming its mucosa. And as you follow that down, you see that all of a sudden this layer of the epithelium in the mucosa seems to diminish. It gets quite a bit thinner. What you're actually seeing is the transition into the stomach, where we're going to find that the mucosa has simple columnar epithelium. We're going to zoom in here to see this a little bit better. Here you can see the nice stratified squamous epithelium of the esophagus, up and down, top to bottom. And right here you begin to see that nice columnar look, single layer of column-shaped cells forming the mucosa of the stomach. Everything you see from here down, you're looking at submucosa, essentially. So that mucosal layer gets a lot thinner in the stomach than it was in the esophagus. Again, here you're not dealing with the same level of abrasion that you did in the esophagus, so that's okay. So on the exams and quizzes, we could show you an image that's zoomed in like this. If the pointer goes here, we'd be asking you to identify one of 2 things. Either we'd ask you for the name of the organ or the type of epithelium the pointer is on. And we could do the same thing pointing down here. And of course, if we show you from 40x or 100x magnification, we'd want the same thing. Identify the organ and or the type of epithelium that you see at the tip of the pointer. So as we move through the stomach, we saw simple columnar epithelium forming the mucosa. The small intestine is going to have the same epithelial makeup. So what you're looking at right here is what's referred to as a villus. This is a finger-like projection. We're going to put its location into perspective here in just a moment. But this is actually going to be a projection of the mucosal layer. And so this is where you find epithelial tissue. Now, like in the stomach, your epithelium of the mucosal or the small intestine mucosal layer is going to be simple columnar epithelium. So it's a single layer of very tall columnar cells. You can see their individual nuclei stacked together like this. And because this is a finger-like projection, that simple columnar epithelium goes this direction here. It's going to curve around and then come back in this direction. And right here in the middle, you're going to see extensions, essentially, of capillaries and then lacteals, those lymphatic capillaries that run throughout some loose areolar connective tissue here through the middle of the villus. Now, what you cannot see quite as well is on the border of each individual columnar cell, you're going to see tiny microvilli. And so by compounding here a structure like a villus, which projects into the lumen, plus adding microvilli on top of these individual cells, you see a large increase in surface area here in the small intestinal mucosa. This is important because this is going to help facilitate absorption anytime you have molecules that undergo chemical digestion and they make their way to the small intestine. Intermittently, you will also find goblet cells. Now, goblet cells themselves look like goblets, so basically this is the top of the goblet where you would see it filled with wine or whatever, and the base of it would be down here in between the individual columnar cells, right? So, or right beside them. You can see it a little bit better right here. There's the base of that particular goblet cell. Those are going to be secreting mucus here into the lumen to help lubricate the surface where food would pass. And so when we take a look here at sections of the small intestine, you need to be able to recognize a few things. First off, you should be able to see that there are 4 different tunics here as you examine a cross-section of one of these small intestine sections. So what you see right here, making up the predominant region, is going to be the mucosa. Right, so this is where you're going to have all those villi, so you can see them here, finger-like projections. They're a little bit smushed, so slides all have different levels of quality depending on how the cuts were made and where. Right? And then below that, you're going to see the submucosa, right? The muscularis, and the serosa. So this is very similar to what we've seen before. Now, the other thing you're going to be able to recognize here is that this is the small intestine, and most specifically, you should be able to tell me that this is the duodenum. It's the first part of the small intestine. So there's a way to recognize this. As you zoom into that submucosal layer right down here, you see a collection of glands known as duodenal, also known as Brunner's glands. And again, those are part of the submucosa. They are unique to the duodenum. So you're not going to see them in the jejunum or the ileum. So when you notice these glands, you'll notice that you are looking at duodenum. That's how you recognize. These glands are helping to secrete a bicarbonate mucus that helps to line the inside of the lumen, preventing breakdown from the acidic kind that you receive from the stomach. When we move to the jejunum, you see again there's similarities here in tunics. Here's the nice mucosa with those villi made of simple columnar epithelium, the submucosa, the muscularis with its 2 layers. Here you can see a pretty nice border between the circular and the longitudinal layer, right there and there. And then the serosa on the outside. You'll notice that there's really nothing extremely distinguishable about the jejunum here at first glance. So the fact that you don't see duodenal glands here within the submucosa lets you know that this is not the duodenum, it is something else. So then if you compare the jejunum here to the ileum, you'll see a number of things about the ileum. First off, in this particular section, it looks an awful lot like the jejunum. There's the mucosa, right? Submucosa, muscularis, serosa. When you look at the ileum from a particular view, one of the things that you'll find is a series of glands called Peyer's patches. So here at 40x magnification, you can see these Peyer's patches. They are associated with the mucosa, and they stain a really nice purple color. Right? The ileum is the only region that's going to have these Peyer's patches. And so if you happen to look at a cross-section of the small intestine and see Peyer's patches, you can know immediately that you are looking at a section of the ileum. If you see, again, duodenal glands and the submucosa right through here, you know you're looking at the duodenum. And if you see neither of those, there's a pretty good chance you're looking at the jejunum. So again, make sure you can identify which region of the small intestine you're looking at, which tunic we could be asking you for. If we're pointing to the mucosa, make sure you can tell us that all along the mucosa of the small intestine you should see simple columnar epithelium, and you should be able to acknowledge any additional glands or structures that are found within these tunics. So of course, as food passes from the small intestine into the large intestine, you see functions change. The large intestine is not primarily there for absorption. Most of that for water and nutrients is done in the small intestine. However, large intestine is going to be dealing with compaction of feces. So these begin to be dehydrated as the large intestine absorbs some remaining water from what's passing through the alimentary canal. And then it compresses all of the waste product to form the feces. That's what compaction is going to be. And so one of the things you notice right here is you'll still maintain that villus, right, those individual villi that you see. You'll see intestinal crypts right through there like we saw in the rest of the small intestine. And then of course you'll see a mass of goblet cells. You can even see all of them right here from 40x magnification. If we zoom in, you get an even better view right there. So more goblet cells than you'll see here in most of the regions of the small intestine. Because the feces themselves are more firm, you do need additional lubrication here along the inside part of the lumen to make sure that there's no problem with anything getting stuck, right? As you would see in situations like constipation. So again, same thing here. Make sure you can identify this is the large intestine. You can also tell us it's the colon. You don't need to identify which region of the large intestine it is. And please make sure that you can identify the tunics as well as cell types and tissue types here of the mucosa. So now as far as accessory organs go, these are going to be organs that are associated with digestive function that do not actually have food pass through them. So the pancreas is review. We actually looked at the pancreas and the endocrine system. What we focused on for endocrine function was the pancreatic islet. So this is where you're going to see insulin and glucagon production taking place. Now we focus on those individual acinar cells, right? Collectively referred to as acini. Those are going to be producing a number of secretions from the pancreas, and then excreting them right here, or secreting them through pancreatic ducts. And those pancreatic ducts begin to converge into one large pancreatic duct that then empties eventually into the duodenum, the small intestine. So while the endocrine function is associated with the pancreatic islets, it's the exocrine function here that's associated with the remaining portions of the pancreas. Here you can see a nice close-up there of those acini. They tend to be a little darker in color with round nuclei. They look a lot like the serous cells that we saw in salivary glands. The liver is a pretty unique slide. The liver itself is going to be broken into a series of hexagonal lobes or lobules essentially that we see labeled here, and you can see really nice clear borders for each one of these lobules. Right? At the center of each lobule, as long as you have a good cut, you should see a central vein. Right? And that central vein is where blood is going to essentially pass through these lobules and then drain on its way to the hepatic vein and returning to inferior vena cava. Now right here is the portal triad. It's named the portal triad because it's going to have 3 different vessels that are associated with it, one of which would be the hepatic artery, and the other is going to be veins that are associated with the hepatic portal system. And then finally you're going to see these tiny little bile ducts. And this is going to give you an idea of the functions of the liver overall. Anytime blood is filtered through the liver, it basically enters a lobule here through the portal triad. The blood will flow through a series of sinusoids, and you can see all those nice light lines right there, like little channels. Those are sinusoids, all the way to the central vein where it's drained from that lobule. Now, as you zoom here to take a nice look at the liver, you can see once again, here's the central vein. You can see the proximity of the sinusoids here, all running into the central vein, like little rivers. And you can see 2 different cell types that we look at here. The hepatocytes themselves are what are responsible for a number of functions, including bile production here within the liver. And so those tend to be the largest cells with a nice round nuclei. In addition to that, you're going to see smaller and less numerous Kupffer cells, right? And these tend to have the pretty compressed and dark nuclei. These are fixed macrophages within the liver, and they have the ability to digest and to eliminate anything that's flowing through these sinusoids here. When we look at the trachea begins our respiratory overview. And so we're actually going to take a look at that in a second video.